Hello everyone. Today we will be learning some things in the subject biology. So what do we have today? We will see today the cell and its environment. What we will be studying in this topic? There are three subparts. First, we will see what is diffusion, next what is osmosis, and then what is plasmolysis. That means that in the topic called the cell and its environment, we will be seeing some basic things about diffusion, osmosis, and plasmolysis. Excited? Yes, let's go. Okay, so before starting this topic, let me ask you one little question. What is cell? Do we know what is cell? Yes, I heard you. You said that cell is known as the basic structural and functional unit of any living organism. Here we have to focus on this particular thing called living organism. Why? Because any living organism, that means we can include any of the plant that we can see around us, any animal, that can be our pet, that can be our cutie, my favorite cat, that can be my favorite puppy, anything, any animal that I see around me, or any of the humans like me, you, your parents, our friends, anyone. So, the cell is known as the basic structural and functional unit of any living organism. Too hard? Okay, let me explain you it in a simpler way. How? Okay, let's take an example of one building, a big one. Yeah. What is it made up of? So, let's first see a big building. Yes, can you see the image of this big building? Okay, now tell me that what is inside the building, how the building is made of. Yes, let's take a guess. Okay, so the answer is bricks. A big building is made up of small bricks. That means if I want to make a big building, I need some small bricks. So I can say that these bricks are the structural and functional unit of any building. The same way, we can apply the same theory for human body. How? So, this is my human body, a little kid who has all these organelles inside his body. He has eyes, brain, mouth, heart, stomach, lungs, kidneys, spleen, liver, intestine, many other things. So, this is a human body. The whole body is looking very good, but it is made up of all these small organelles. And these small organelles are made up of simpler types of cells. So, these are the types of cells that we can see in our human body. They can be of various types. They can be of stem cells, they can be bone cells, they can be blood cells, muscle cells, fat cells, skin cells, nerve cells, endothelial cells, sex cells, pancreatic cells, and cancer cells. So, what I want to see is that if I'm considering a human body, then I must say that a human body contains many types of organ. And each organ is made up of cells. Right? So this is how we can say that cells are the building blocks of our human body. Getting my point? Yes? Okay, so now let's move further. Now, this is the thing that we know since our childhood. And I just cleared this thing when we started the lecture. What I said that any living things can be a plant, can be animal, can be humans, right? So now, cells can be of two types. Which two types? So first are plant cells and second are animal cells. Let's first talk about plant cells. So this is a figure of plant cell. So what can we see in this plant cell? So before we understand something about plant cell, let's first see a figure of animal cell. Then we will be able to differentiate between these two cells. Okay, so this is the figure of an animal cell. Now tell me, what do you see in these cells? Yes, you get it. This is a plant cell, and this plant cell has something like this in its outer border, while the animal cell doesn't have anything kind of this. So this is the structure that is called cell wall. That means plant cells possess cell wall, while animal cells possess no cell wall. That means no cell wall is present in any animal cell. Clear? So depending on the presence of cell wall and the absence of cell wall, the plant and animal cells functions a little different way. How? So the cell wall makes sure that the plant cell stays rigid. It doesn't change its shape. While in animal cell, if something happens with animal cell, it may change its shape. Okay? Now, any living cell will have to interact with its outer environment. Now, when I say to you that a human body is made up of different types of cells, so these cells, we are living beings, right? We can live, we have to go for nutrition, we have to breathe, we have to get our food, we have to get oxygen, we have to take out carbon dioxide from our body, we go for any other thing, we go for playing, we go for crying, anything. So in simple ways, any living cell, I'm talking about only living cells here. So any living cell will have to interact with its outer environment for different purposes. Which purposes? So it can be nutrition. Nutrition means the food that we eat that provides us energy so that we can do our things. If I can't eat, then how will I play with my friends? Is it possible? No. I have to have my favorite cakes or my favorite burgers so that I can feel energetic and I can go to school and play with my friends. Yes, so this is nutrition. Next is respiration or gaseous exchange or gas exchange. So we breathe in, we breathe out, right? You can do that right now. You can just breathe in once. You are taking in oxygen and breathe out. You are taking out carbon dioxide from your body. So this is the gas exchange that is occurring inside your body. And the whole process of breathing in and breathing out is called as respiration. And the last is excretion, right? What is excretion? We go for loo, we go for washrooms, right? So this is excretion. We sweat, we pee. So in this way, we remove the excess or extra materials which are waste products in our body. We remove those waste products from our body in the outer environment. And all these processes occur inside a particular cell. So that's why we can say that any living cell will have to interact with its outer environment for these different purposes. Okay. Now tell me, what is concentration? Do you know what is concentration? Yes? No? Okay, fine. Let me explain it. For understanding concentration, let's take an example of a glass and a salt which we are adding into it. Yes, can you see this glass? In this glass, I have added some amount of salt. So, imagine that this glass has no amount of salt in it. That means that this glass has only pure water. Now, when I add salt into it, that means that I am adding some amount of solute into solvent to make a solution. Clear? So in this glass, when there was pure water, it was a solvent. Now I added some amount of solute or salt inside. That means that now I can say that this amount or this much amount of salt is inside this water. Now, this particular amount is called the concentration. That means if I have added 2 grams of salt in this water, I will say that the concentration of this water or the salt inside the water is 2 grams. Clear? So now we will see, or we will move on to our basic topics that we will be seeing in this chapter. The first one is diffusion. What do we know about diffusion? Let's see. So, as you can read here, diffusion is the movement of molecules from a region of higher concentration to a region of lower concentration down the concentration gradient. So, just before 1 minute, we just understood what is concentration. That means if something is dissolved in a solvent, one solute is dissolved in a solvent, that means that is the concentration of that solute in the solvent or solution. So, diffusion is something which says that a molecule which is present in the higher concentration region will move to the lower concentration region down the concentration gradient. Too hard, no? Let's take an example and understand it in an easier way. Okay, so here you see 2 glasses. In one glass, this glass, I have just added a blue colored salt. So when you see, when I poured the salt inside the glass, the salt is here. The higher concentration of salt molecule is here. Now, due to the diffusion says that due to diffusion, the molecules will move from a region of higher concentration. Here the concentration of the salt is high, so the molecules will move from a higher concentration to a region of lower concentration down the concentration gradient. That means here in the bottom part of a glass there is no salt concentration. So the salt will move from its higher concentration to its lower concentration. So it will move in this way. So what happens when the salt moves in this way? This happens. So the salt is dissolved in the whole water. The colored compound which was initially only present here, now it can be seen inside the whole glass. So this is the phenomenon of diffusion. I hope that you got it, right? Okay. Now, when a living cell is considered, they have permeable cell membrane. What is permeable cell membrane? So permeable cell membrane is something that allows a cell to enter only few things. How? So the same way, when your mom serves you your favorite food, if it has chocolates, if it has cakes, and if it has a pastry, then if I like the cake, I will only tell my mom to put only cakes inside my plate, not any other thing. The same way, the permeable cell membrane that is present around the cell will only allow the entry of particular or few molecules and not every molecule that is around the cell. This is to protect the cell from the harmful effect of all the other chemicals or things which are present outside the cells. Okay, so now this permeable cell membrane controls the movement of substances. So this plays an important role in diffusion, because we say that diffusion occurs when the molecules move from its higher concentration to lower concentration. Now, the uses of diffusion can be where we can see the use of diffusion in animals and plants. So, there are 3 possible events when diffusion is involved in humans, animals or plants. The first is to obtain nutrition. When any animal or human or plant wants to obtain the nutrition inside the cell, it will move with the means of diffusion. Next is for gaseous exchange, that we discussed earlier, that when we breathe in, we take in oxygen inside. When we breathe out, we take out carbon dioxide from our body. So this is the gas exchange. Where does it happen? It happens in the alveoli of the lungs. So, and the last is excretion. Excretion means the waste product which are generated in our cell. We will remove those waste products from our cell. So this is the figure where you can see what exactly happens during the diffusion. So if the cell requires nutrition, then the food molecules will go inside. If the cell is supposed to do the gas exchange, then the oxygen molecules during the breathe-in condition will go inside the cell. And if a person is breathing out, then the carbon dioxide will come out from the cell. All these things happen with the means of diffusion, right? Now, I want to tell you one thing that you should remember for your examinations. This can be a very good question. What is that? A diffusion is not energy demanding process. It doesn't demand any energy. That means that there is no requirement of energy during the process of diffusion. Clear? Okay. Now, let's see what is osmosis. So, we can say that osmosis is a movement of solvent particles from its higher concentration to lower concentration across semi-permeable membrane till the equilibrium is achieved. Now, what is the definition trying to tell us? So, first, osmosis. So, in osmosis also, there will be a movement of molecules or particles. But particularly here in osmosis, only solvent particles will move, not solute particles. Now, the solvent particles, that means if I take the example of that glass where I took the salt plus water, so here my water will be my solvent and my salt will be my solute. So only solvent particles, that means only water, if I consider this example, the solvent particles will move from its higher concentration to lower concentration across the semi-permeable membrane. We just saw what is permeable membrane. So permeable membrane is a membrane which allows the entry of only particular molecules or compounds. The same way, semi-permeable membrane is a membrane that allows the entry of only particular or defined or predefined molecules which are defined when the permeable or semi-permeable membrane was made. So in simple language, we can say that in osmosis or in the process of osmosis, the solvent particles will move from its higher concentration to its lower concentration till the equilibrium is achieved. Now, what is equilibrium? So equilibrium is a condition where if I say that one person is having 4 chocolates and other person is having 2 chocolates. So they are not at the condition of equilibrium. One has more chocolate, other has less chocolates. So let's ask this child to give 2 chocolates to this one. So now what will happen? They both will have 2 chocolates. That means now they are at the equilibrium. That means when both the solutions are at the same condition, they will be called at the condition of equilibrium. Getting my point? Yes? Okay. Now, let's try to understand this with a figure. Yes, this is a semi-permeable membrane. That means that this membrane allows only the entry of some particular compounds and not all the compounds. Now, here is the high concentration of solute. That means the salt concentration, if I take this example, then the salt concentration is higher in this side and lower in this side. So due to the phenomenon of osmosis, what will happen? We said that the solvent particles will move from its higher concentration to its lower concentration. So water will move towards the high solute concentration. That means here the solute concentration is high. So the water molecules are bound with solute. So the available free water is in less amount. So the high solvent particles are on this side. So what will happen, that the solvent particles will move from this direction to this direction. Now the both solutions are at equal concentrations, right? So this is the phenomenon of osmosis, which says that whenever osmosis will take place, a solvent particle will move from its higher concentration to lower concentration till the equilibrium is achieved. Okay, great. Now, we can say there are three possible types of conditions which can occur in the outer cellular environment. Which three types of conditions? So let's see that. Yes, there are these three types of conditions. First is hypertonic, second is isotonic, and third is hypotonic. Now, just in the previous slide, I mentioned something about equilibrium. Equilibrium means to have the equal concentrations. So let's start with the isotonic concentration or the isotonic condition. So when I talk about the isotonic condition, I say or I mean that when a cell is placed inside something which has the outer environment which has the same constituents or which has the same amount of solutes and solvents as the cell. So there is no change inside the cell or there is no change outside the cellular environment. In simple words, if I say something about isotonic solution, then I can say that there will be the same condition for cell inside and outside. Right, there will be no concentration difference. So there will no water or no solvent particles will go inside, no solvent particles will come outside. Now let's talk about hypertonic conditions. So what happens during the hypertonic conditions? So during this time, the solute concentration is higher outside the environment. That means that solutes are more in outer environment or in outer side. So due to this, what happens? We just saw that due to the process of osmosis, the solvent molecules will move from its higher concentration to lower concentration. Now, when the solute particles are higher inside the outer environment, the water from cell will come out. So due to this, our cell will shrink. You can see this in the image. Okay. And the cell which has undergone this process is called a plasmolysed cell. Now, when we go for isotonic condition, so the cell doesn't face any difficulties and it is in the same condition. So that cell is called as flaccid cell. Now, the last condition, that is hypotonic condition. So what happens in the hypotonic condition? So the outer environment where the cell is placed, it contains the less solute amount than inside the cell. That means in hypotonic condition, solutes are less. So what will happen? Due to the phenomenon of osmosis, the solvent molecule or the water will move from its higher concentration to lower concentration. Inside the cell there are high amount of solutes and outside the cell there are less solutes available. So the free water is more in the outer environment. So the cell will face that high amount of water will go inside the cell. Due to this, the cell will swallow, and this kind of cell is called as turgid cell. So when we study osmosis, we have to deal with these three kind of solutions or these three kind of conditions that can possibly occur around the cell. They are hypertonic, isotonic and hypotonic. Okay. Now, if I want to understand osmosis, then I can understand this whole process by the example of three things. First is blood cells that are already shown here. What is that? So this is the isotonic condition for the blood cells. What happens here? So here the blood cells are put in the environment which is the same as the cell. So there will be no high water will come inside or no high amount of water will go outside. That means that cell will be at equilibrium condition. So this is the isotonic condition where we can see that blood cells are in their normal condition. Now when we go for hypertonic solution, when the blood cells are put in an environment which has high solutes in outer environment, then what will happen? We just saw it in the previous slide. The water from the blood cells will go out, right? This is already shown here. So due to this, our blood cells will shrink. And when we go for hypotonic environment, what will happen? The solute concentration is lesser in the outer environment. So the solvent molecule or water molecule will continuously come inside the blood cells. So the blood cells will swallow and it may burst. You can see it here. Okay. So this is the first example by which we can understand the phenomenon of osmosis. Next is grapes. You can do the experiment. You can take the grapes and you can put it in hypertonic solution and hypotonic solution and in the isotonic solution. Okay. And then tell me what is your results. And the last is raisins. So with these three examples, we can understand the process of osmosis. Now let's understand the phenomenon of plasmolysis. What is plasmolysis? So plasmolysis is called as a process in which cells lose the water in hypertonic solution due to the exosmosis process, and due to this the cell shrinks. So it is shown in the figure what happens. Usually it is seen in the plant cells. So as you can see what happens, this is the normal cell, right? This is the cell wall. Now when the cell is put in an hypertonic solution, what will happen? The solute concentration will vary from the cell's normal concentration. The solute concentration will be high in the outer environment. So the water from the cell will go out. So as you can see here, plasmolysing solution. So the cell will shrink due to the loss of water. The water from the cell, this is the pure normal cell. Now the cell is losing its water. And when the cell has lost almost all its water, it will shrink like this. So this is the process of plasmolysis which occurs when we put a cell into a hypertonic solution. And due to plasmolysis, the cell will get shrinked. Okay. As I just mentioned, the process is usually seen in plant cells. And as the cell wall is present in plants, the cells just get shrinked during the plasmolysis process. And once they are again in hypotonic solution, due to the process of endosmosis, they will again get back into the shape. That means that when I will put a shrinked cell inside a hypotonic solution, that means the solute concentration is less in the outer environment, the water will go inside the cell and the cell will get back into its normal shape. So this is the process of endosmosis where the water goes inside the cell. Right? So what we can say? Endosmosis, in this process, the water will go inside the cell, and during the exosmosis, the water will come outside from the cell. So this is one of the examples of a plasmolysis. In this condition, this is the normal cell, and when the cell was put in the hypertonic solution, this happened to it. So you can see that the cell is shrinked, but due to the presence of cell wall, There is no damage to the cellular structure. Okay, so that's all from my side. I hope you understood well what is osmosis, what is diffusion, and what is plasmolysis. Thank you. Take care and stay safe till we meet again. Bye.